A large nose and black curly hair, brown eyes and bushy eyebrows joined, two big ears make him round, rounder. That's your 23 pairs. Now you're done. 1, 2, 3, 4, 5, and 6.
Mosquitoes have 6. Bananas, 22. Flies have 8. Chimpanzees, 24. Cows, 30. Chickens, 39. And butterflies, 190 pairs.
That's the number of chromosomes. Numbers, it's all in the numbers.
Numbers and codes, genes and chromosomes, the DNA and its twist. Genetics is a complete science. The Biotechnological Revolution Gallery at the VITM Bangalore makes the discovery of genetics fascinating. Genetics, an important part of biotechnology. It's an exploration that begins with the cells. More than 10 trillion cells form the human body. 10 trillion. Their sheer variety is amazing. Nearly 210 different types. The cells look different. Their functions are different. Shapes and size and lifespan. Even that is not the same. Together they make different tissues and organs. But whether in the heart, in the muscle or the brain, the trillions of cells in any body share their DNA, the unique blueprint. They belong with each other. At the gallery, one can see how it is. In each of the cells inside any one body lies a nucleus, and inside the nucleus are the chromosomes. Chromosomes made of DNA that carry the information. At the cellular level, this is the scheme. The nucleus, and inside it, the genes. More than 30,000. Small parts of a very long molecule, the DNA, deoxyribonucleic acid, the twisted molecule.
DNA, the twist of life. The blueprint unique to any individual. And here, I just don't mean you and me. I mean just any individual.
A sheer variety of life fills our world. Plants, animals, bacteria and fungi. Each life form unique. Let's take one family. All cats, but each very different from the other. A cat at home is a friend. But try getting a lion. The difference in strength and behavior, size and characteristics lies in their specific genes, their DNA. So each member of the cat family is similar yet different. Similar because they share more than 99% of their DNA. Different because there is a minuscule difference. Chimpanzees and humans share 99% of their DNA, yet man is man and chimps, chimps. At the gallery, one discovers fascinating facts. Man shares 99% of its DNA with the mouse, 46% with the humble E. coli, 50% with the fruit fly, and 40% with the nematode worm. That's a lot of sharing between such different species. All humans share 99.8% of their DNA, yet each is unique.
So how such a small difference can make such a big deal? What is the mystery behind the twisted ladder, the DNA?
A long, double-stranded molecule like a twisted ladder. The DNA is made of sugar, phosphates, and 4 different bases: adenine, thymine, cytosine, and guanine. The rungs made of these nucleotides: adenine paired with thymine and cytosine paired with guanine. Just 4 bases and always in the same combination. It's an elegant, logical design. If these are the 4 nucleotides, one can see that adenine can only join thymine and not guanine or cytosine. And cytosine and guanine can only pair with each other.
Tall and long. Just how tall?
As tall as Amitabh Bachchan. A single DNA uncoiled is about 6 feet long.
Wow.
So how do you fit something that long into something smaller than a pinpoint, the cell nucleus? This is exactly how. The DNA coils tightly and forms a chromosome. It becomes so small that it easily fits into the microscopic nucleus. The interactive computer-based model makes the structure clear. Here's the coiled chromosome inside the nucleus, the 2-armed chromosome made of tightly coiled DNA. Let's imagine the nucleus of a human cell. If this is the nucleus, a tiny fraction of a microscopic cell, then 46 chromosomes fit inside. At the Biotechnological Revolution Gallery, one discovers the amazing unity in diversity. Man shares its recipe for life, its DNA, with all other life. Be it the insects, mammals, birds, or even the grasses and trees. Not the number of chromosomes, not even the length of the DNA. Then what is the secret of this code that makes each species unique?
Let's go back to the DNA.
At the gallery, one understands that it is the DNA that tells anybody how to grow. It is the instruction manual for building a body. In similarity lies amazing diversity. Can you identify the human fetus? They look so similar. But one is human, the other a chicken, and one a fish. It is the chromosomes that hold the key. Chromosomes, unique to any species. Chromosomes that carry the genes. Genes that can be passed down generation to generation. Genes carry the blueprint from the parents to the baby. It is the genes that ensure that like begets like.
Let's play a game of word building with just 4 letters: O, S, P and T. But that's just 4. 4. Nalaku. Now let's think about this.
Genes are also made of just 4 alphabets, only 2 base pairs. But these 2 pairs combine in various ways and code for the 20 amino acids. Amino acids, vital to life, the building blocks of proteins. These base pairs form millions and millions of different combinations, a whole dictionary of very diverse life. A gene is a stretch of the DNA, a combination of 3 base pairs. It's called a codon. Different parts of the DNA have different codons, a set of instructions that dictate different traits in the body. Physical traits, even blood group, or the body's ability to fight disease. Just about everything is controlled by a gene or a set of genes. Genes tell the cells how to function and what trait to express. We can think of them as recipes for making proteins. Different parts of the DNA or genes get activated. Their code gets copied and instructs the cell to begin production. Let's travel inside a hair follicle. That's where the protein keratin is made. Keratin that leads to hair growth.
Keratin, more keratin, and some more. Let the hair grow.
40,000 different proteins is what we need every day for life. It is the genes that give orders and the cells follow. They link amino acids together to make the proteins. A protein chain 400 amino acids long can be assembled in just 20 seconds. The codons carry the recipes, and there are even start and stop codons. Almost like switches that turn on and off. And regulators that increase the pace of production or slow it down. The DNA has it all encoded in its 4-letter dictionary. At the gallery, one can learn and put the knowledge to test. It's an interesting interactive model for protein synthesis. A single click to unzip the DNA that lies inside the nucleus. At every step, one understands how it happens and why. Once unzipped, the messenger RNA strand appears. It has 9 bases. One has to choose the right base and complete the pairing. Each time, adenine joins thymine and cytosine pairs with guanine. Remember, in a real cell, the RNA strand could be 100 to 10,000 bases long. And inside the cells, this process happens at an amazing speed. This part of the process is called transcription. Once ready, the messenger RNA moves away from the DNA. It leaves the cell nucleus. Outside, the ribosomes attach themselves to the messenger RNA. One can drag the tRNA with matching amino acids onto the mRNA and form a polypeptide chain. Meet Papa, Mummy. Then came Rohan and Rajita, siblings born of the same set of genes, yet different. But why?
Literally that. But how does it work?
The gallery makes the process easy to understand. Let's start the exploration with James Watson and Francis Crick. The DNA had been known much before their time, but they were the ones who figured out the double helix. They even made a 3D model of the DNA and cracked its code and structure. Watson and Crick were brilliant, and so was Rosalind Franklin. She unraveled the biochemical mysteries of the DNA, the knowledge that led to a rapid growth in the understanding of genes and hereditary. Every child carries his parents' DNA, 23 pairs of chromosomes inside each cell. For this to happen, the parents need to contribute just half their DNA. That's what the sperm and ovum have, just 23 single chromosomes. When these two sets meet, they form pairs, two strands containing the same genes but different recipes. They combine and exchange sections, a random process that creates a brand new code, a complete set of 23 pairs for the baby. The child begins as a single cell with its unique code and soon grows. Each cell in the child's body with its unique DNA for life. But what determines the sex of the child? It's a matter of chance. It depends on which chromosomal strand of the father unites with the mother's. It's the 23rd chromosome. A girl is a girl because the 23rd chromosome gets the X strand from the father. A boy is a boy because his 23rd chromosome has the X strand from the mother but the Y strand from the father. The sex of the child is a biological process in which the mother has no role to play.
Aha! Just a matter of chance.
So, are genes like watercolors that mix and create new shades? Or are genes like beads that string together to form unique individuals? How does the child get some features from the father, some from the mother, and some even from the forefathers, uncles and aunts? It's like mixing two packs of cards.
I now call upon the father of genetics. Hail the king!
Gregor Mendel. He was intrigued by how traits were passed on and expressed. With his famous experiments with peas, Mendel learned that some traits were dominant and others recessive. Let's understand what Mendel understood. Capital letter for dominant gene, small letter for the recessive one. When Mendel crossed yellow round peas with green wrinkled ones, he found that the yellow and round traits dominated, but even the green wrinkled showed up. Was it a game of chance? The father has the gene for blue eyes, the mother brown. Balls are the genes, red for brown, blue for blue. One red, one blue, the child would have brown eyes. Two reds, brown again. But if the child catches both blue balls, then the eyes would be blue. The study of heredity has been a slow and steady process. The brilliance and hard work of several stalwarts has added to the knowledge. Over time, it became known that certain traits tend to dominate. When it comes to the color of the eyes, brown eyes dominate over blue. The ability to roll the tongue dominates over the inability to roll the tongue. And if the child inherits the gene for extra fingers, then that dominates over 5 fingers. So genes are really like beads that string together to express different traits. The traits cross over, recombine, and create unique individuals each time. How individual? Let's check the genetic wheel. It's a fun activity. You have to see yourself in the mirror and match your features. From the dimples in the cheeks to the ability to roll the tongue. Time to check. The only one. The rules of genetics, of recessive and dominant traits, of recombining and crossing over, hold good for all life forms. In diversity lies a unique similarity of mechanism. The gallery arms the visitors with knowledge and then offers them an opportunity to check it. Identify the parents checks one's understanding of heredity. We know that genetically related persons have some bands in common with their DNA fingerprints. So looking at the child, can one find out the father and the mother? It's a fun way to learn. The display even tells you if you've got it right.
But there is another twist in the story. What if neither of the genes is willing to be dominated?
Blood is a great example, a complex living tissue that contains many cell types and proteins. Humans have 4 basic blood groups on the basis of the antigens present. Why 4? The secret lies in the genes. A group has the dominant gene for antigen A. Those with blood group B have the gene for production of antigen B. In O there is neither, making the person a universal receiver. But for AB group, it's codominance in action. The blood has both the genes for antigen A as well as B.